And then I'm going to go ahead. All right, everyone. Hi, welcome. Thanks for coming for another week of Conservation Sciences seminar. Today we have Dr. Scott Colborne coming and joining us virtually from Michigan. So he is originally from Southern Ontario and received his bachelor's and master's at the University of Guelph. I would not have pronounced that correctly. Thank you.
You're welcome.
Followed by a PhD at the University of Western Ontario. His graduate research was focused primarily on foraging diversity within and among bluegill and pumpkin seed sunfish from lakes in Eastern Ontario. He completed his postdoc research projects at the University of Windsor and a joint USGS Michigan State University position. It was during this time that he began to work with acoustic telemetry to track fish in the Great Lakes region. Projects during this time included a large-scale tracking study of Lake Sturgeon in the Huron-Erie corridor, and he spent 1 year with the Shedd Aquarium on their conservation research team before moving to a postdoc with University of California, Davis. It's the acoustic telemetry work in the Central Valley of California that will be the focus of his seminar talk. And in December of 2022, he started a research scientist position with the Quantitative Fisheries Center in the Department of Fisheries and Wildlife at Michigan State University. In this role, he's continuing to work on a variety of telemetry-focused research projects in the Great Lakes region and to better understand the ecology of freshwater fishes and inform conservation and management efforts. And with that, I'll turn it over to you.
Thank you very much. I know I gave you plenty of text for that intro, but you got through it just fine. And there we go. So as Olivia said, I am currently with Michigan State University and the Quantitative Fisheries Center, but I started that position just a few months ago. And the work I'll be talking about today actually covers the 2 years before that, where I was doing a remote postdoc with the University of California, Davis, on using telemetry that's been done in the Sacramento and San Joaquin River systems of the Central Valley, so up in Northern California, to better understand the ecology and inform management of freshwater fish. To start off, I definitely have to acknowledge the huge number of partners that were part of this research. I came into this project when data was already, in many cases, more than a decade into being collected. We have various groups and organizations. I worked directly on a team through UC Davis that involved the University of Virginia and the University of Kansas. But we had funding and work partners across all levels of the state and federal, from the Delta Stewardship Council and California Department of Fish and Wildlife to U.S. Fish and Wildlife Service, USGS, the Bureau of Reclamation, which is a huge partner in the water research in California, more than I think most people in this region, in the Great Lakes region, think about, and NOAA as well. Freshwater is a critically important part of the entire global ecosystem. It may represent less than 1% of the total planetary water supply, but more than 140,000 species depend on access to fresh water, either as where they reside for their entire lifetime or for parts of their life history. And this represents 12% of all described species, despite being 1% of the water supply. Also, freshwater is important. These species are important for humans, both culturally. They can have major significance to different cultures around the world. And also are a critical part of food systems for populations on every continent. Unfortunately, freshwater populations have declined an average of 84% since 1970, and it's estimated that about 1/3 of freshwater species are currently at risk of extinction within the next century. And this extends to where my work in California comes in, because freshwater is a vital part of California and its ecosystems, even though we tend to think of it more as being a coastal state, so the Pacific would possibly be given more focus. But freshwater is absolutely vital in this region, especially Northern California, where freshwater is contributing to actually producing 25% of the total national food supply for the United States. We don't really tend to think about California as an agricultural powerhouse, but it actually is. And there's more than 250 types of crops growing in California that provide food to the US population and, quite frankly, people around the world. In particular, the Sacramento and San Joaquin river system up in Northern California is a freshwater hotspot. It receives 40% of the total precipitation in California falls into those river basins. And it has 50% of the total river flow moving through the state. More than 50 fish species reside in the Sacramento and San Joaquin rivers. There are many native species. There are also quite a few invasive species, including some that we would find surprising because some of the native fish from here in the Great Lakes region are now problematic invasive species on the West Coast. There are, unfortunately, though, many of these native species are at risk of extirpation and extinction for a variety of reasons. Probably most famously would be some of the salmon, Chinook, and steelhead being key examples that I'll be talking about later today as well. Why are there conservation challenges for fish in California? Well, there's many different reasons. Some it's directly from overfishing. We have, when fish are in these rivers systems, we tend to have better access to them, and so we can catch more than was sustainable. Hopefully those habits are changing with stock assessments and those regulations. Another major one for across California are blocked passages and water diversions. So here's an example of a dam up on the upper Sacramento River system. But water diversions are also a major issue because, how I mentioned, that 25% of the crops for the country are growing in Northern California or in California. To get the water for that agricultural development, there's diversions directly pulling from a lot of rivers and lakes. This has also been exacerbated by habitat loss and drought. California has just gone through a several years of drought conditions in a row. We've been hearing a lot this year about the entire Western US having major issues with water levels dropping, and California has most definitely been part of that. They're getting a little bit of a respite this year, but we don't know how long that will continue. And I mentioned briefly before invasive species. This is a picture of a largemouth bass. We think of them as native in this area of the world around the Great Lakes, but they're actually a problem invasive species on the West Coast because they have taken up residence in a lot of these river systems, and they can be very good at finding the salmon smolts when they're young and being major predators for them. Now, my research has tried to address ecology questions as well as conservation and management issues in California using acoustic telemetry. And I'm sure some of the people here will be familiar with acoustic telemetry, but for those of you that aren't, I'm going to take a couple minutes and actually just show you an animation explaining it from the European Tracking Network. And this is mostly marine focused, but the principles apply to what we're doing here in freshwater as well.
Aquatic ecosystems are full of life. Atlantic salmon migrate in and out of rivers to spawn. Sharks can move long distances in the ocean. Lobsters move around the ocean floor foraging. Eels migrate incredible distances to spawn at sea. Sunfish roam the wide ocean. Tunas can dive down to more than 1,000 meters. Understanding where, when, and why animals move is necessary to adequately protect and manage them in a sustainable way. On land, we can use GPS to track animals, but that doesn't work in water. So how can scientists get this information when animals live underwater? One way to do that is using acoustic telemetry. Telemetry means to measure from a distance, and acoustic means using sounds. So Scientists use acoustic telemetry to track animals from a distance using sounds. Once the study animals are caught, they undergo a small surgery so the scientists can implant acoustic transmitters. For larger fish like sharks and tunas, or for animals with a hard shell like lobsters, the acoustic transmitters are attached externally. These transmitters emit a sound through a sequence of pulses. The sequence is unique for each tag, so each animal has a unique ID. The IDs can be detected when the tagged animals swim close to receivers, as the receivers detect the sound emitted by the tag. Researchers have a network of thousands of receivers across Europe to track aquatic animals. The receiver information provides data on when and where animals move, but it can also assist scientists in understanding why they move, such as for spawning or feeding, or even figure out where they are seeking refuge. The tagging research can help managers in decision-making processes, like where to create marine protected areas or MPAs, to remove barriers for movements, like dam removals. In this way, acoustic telemetry is an important tool to inform the sustainable management and stewardship of aquatic animals by providing knowledge on their movements, habitat use, and survival, particularly in the face of changing global environments.
So yeah, even though that was focused on a marine application, because much of the work done by the European Tracking Network is focused on the oceans and the Mediterranean Sea, the principles, the types of questions, the applications, they hold in freshwater systems too. And so that hopefully that gave you a good intro to what acoustic telemetry is. But just to sum it up with a couple key points, the type of acoustic telemetry that I've been doing, it is passive monitoring of tagged animals, and it can be done from small to large sizes. So I'm going to start today by talking about large adult green sturgeon, some of the largest fish found in the rivers in California. And then we'll be talking about Chinook and steelhead smolts, some that are barely the size of your forefinger. These transmitters will ping at known intervals, so we know roughly how often they should be transmitting. And they do provide long-term tracking of fish as long as they pass within the detection range of a receiver. That being a key limitation, or at least a key variable in the success of acoustic telemetry projects. If the sound is being transmitted away from a receiver, we'll never know where that fish was. So they have to come close enough to be picked up. And so acoustic telemetry has become a focus of my research and trying to understand what we can learn about fish ecology to inform conservation management using this tool. So the first case study that I'm going to talk about today is from California, is the migration of green sturgeon in the Sacramento River. There are two species of sturgeon native to California, white sturgeon and green sturgeon. The white sturgeon they can grow a bit larger than the green sturgeon, but more in their northern part of their population, up along into Oregon and Washington and British Columbia. In California, the green sturgeon tend to actually be the larger of the two species. And for this project, I focused on detection records in the Sacramento River. So for those of you who don't know, the Sacramento River runs through a region of California and all the way up into Oregon. It starts, though, in an area of California known as the Central Valley. And this connects to the Pacific Ocean through a series of bays and the Sacramento-San Joaquin River Delta. Most famous landmark that we could possibly use as an anchoring point is the San Francisco Bridge. These green sturgeon, in order to get out to the Pacific Ocean, that's where they're passing through. They're going right under the Golden Gate Bridge in order to get to the ocean. And the Sacramento River itself is home to a diverse array of fish. They range from resident species that spend their entire lifetime in freshwater to others that migrate for various periods of their life. Probably most famously, you can think of salmon. There's multiple salmonids that migrate in and out of the Sacramento River, but they absolutely require access to this freshwater system in order to spawn and have the first stages of juvenile development before they move out to the ocean. For green sturgeon, the population in California is known as the Southern Distinct Population Segment. It differs from the population further north along the coast of Washington and Oregon. They were actually listed as threatened under the Endangered Species Act. So that means that there are special protections and regulations in place for these animals, including a mandate to better understand what they need from the river system. General movement patterns have been described from traditional methods, mostly by catch and release trapping. And in general, what they have described is that in the spring months, the green sturgeon adults will come in from the Pacific Ocean and move up the Sacramento River, moving as far as 400 kilometers up to spawning grounds. They will spend a few months there, and then they come down during the late summer or early autumn. That's the traditional view of their migration. But there were questions that were raised about how accurate this was. And this is where telemetry came in to being a tool to try and assess this, because the timing of movements for sturgeon as a threatened species can really impact the water diversions, operation of dams, and seasonal barriers. So multiple agencies have tagged green sturgeon over more than a 15-year period now to better understand the adult use of this system. So the project objectives that I had coming into this were to describe the timing of up and down river migrations of green sturgeon in the Sacramento River. And the specific question that we wanted to get into was whether the green sturgeon migrate upriver during a relatively short period of time each spring. So basically, is there one pulse of upriver migration, which based on the typical spawning activity that we see in other sturgeon and freshwater fish in general, we predicted that there would be a single period overall where most of the fish were moving upriver. Then we wanted to ask if there were multiple downriver migrations. And one of the reasons, in addition to it mattering for water diversions and management of the system, we also thought that we had a prediction that there could be multiple downriver migrations, because further north in the northern distinct population of green sturgeon, this has also been observed. So where there are some adults that will spend an extended period of time in the rivers before they come down, and others that basically spawn and head downriver right away. So we didn't know for sure with the southern population. So it was possible that they either had multiple peaks of migration or they would all come down at roughly the same time. So how did the telemetry data came into this? We used the PATH database, which is an acoustic telemetry database housed at UC Davis with many partners contributing to it. And we queried the detection histories for 350 tagged green sturgeon from 2006 through 2018. And there were 151 fish that were present in the Sacramento River during this time period. So we focused on those green sturgeon. And we collected all of the detections from more than 300 receivers, starting from the Pacific Ocean and the Golden Gate Bridge, all the way up to just south of the Keswick Dam, which is a barrier that they wouldn't be able to pass. And so this figure on the right, each of the dots shows you where a receiver was for at least part of that time period. So you can see we had pretty good coverage throughout this entire area. We considered upriver migrations to begin once a green sturgeon left the inland delta. And just to give you some context, this area down here in black, this is considered the delta for this area. So once they left the delta and were heading up the main part of the Sacramento River, we considered this the beginning of their upriver migration. This distinction was made because it's not uncommon for adult green sturgeon to basically come into the delta for foraging. But once they go up the river, they're heading up for spawning. We don't have any evidence that they head upriver 400 kilometers without spawning. Then the purple part in here is what we call the migratory river run. Basically, this is a time where they're just moving as quick as they can up or down. They're not stopping in many places. And then up in this yellow area further north, this is where we tend to see holding and spawning activity. And so once they reached this part of the river, anything above here, they were considered to potentially be within the spawning area. And then conversely, when they were coming back downriver, as soon as they moved below into these purple areas, we considered that the end of their spawning period and the beginning of their downriver migration. So we, going through all of the detection data for those 151 individuals, we found 129 upriver migrations and 224 downriver migrations. And if you're like me, when I first started working on this project and you saw these numbers, you immediately went, wait a second, why the difference? Why would we have almost double the number of downriver migrations? And then it made sense when we looked at the tagging history of these fish. It's easier to find and capture green sturgeon when they're in the rivers. So the majority of fish were tagged when they were already partway up the river or already at near the spawning grounds. So we weren't able to get a good record of the upward movement, but we had the downward movement. We also wanted to look then if we could find paired detection events. So those times where we got the full upriver migration and then the full downriver migration following. And for that, we identified 117 full migration events. And this is an example across 6 years of what that looked like in these waterfall plots. So zero is zero on the y-axis. That's the Golden Gate Bridge in San Francisco. That was our 0 point for river kilometers. And then they would move up through the delta, up the river, and then up in this area, the 400 to 500 river kilometer range, that's the spawning grounds and the holding area. So you can see that in general across all of these years, there's some variability, but basically what seems to happen in the spring, fish move up quite quickly. They spend time up there, and then some come down fairly soon after. Others hold over for several months and then come down. In general, the upriver migrations occurred from February to May of each year, with the mean being mid-March or late March, and it happened with a unimodal peak. Temperatures typically were about 15 degrees Celsius, and then it was also at fairly high discharge levels in the river system. Downriver migrations spanned a much wider period, which I'm sure people looking at these waterfall plots already are seeing, that it went from April all the way to January. So some fish spent almost a year up the river before they came back down. In general, the mean across all of the fish was October 16th. So that late summer, early autumn period. But there were two distinct peaks here across. I'm showing 6 years on this panel, but we actually had more than 10 years that we were looking at. And this pattern held across that there was an early peak and then a late peak and a period in between where very rarely did any of the sturgeon go downriver. So what about the timing of those two peaks? How did they differ? Well, early downriver migrations happened in June, mid-June. The late ones happened in mid-December. They happened at fairly different temperatures, but was really and then also the discharges were much lower than during the upriver migration period. We also saw that about 1/3 of the events were in the early group and 2/3 were in the late group. So there seems to be there seems to be a tendency for the green sturgeon to hold over for the better part of a year before they come down. And we wanted to ask if discharge or temperature were related to green sturgeon starting the downriver migrations. And so for this, we did a time to event analysis using Cox proportional hazards regression And we did this separately for the early and late groups. So fish were classified into one of the two groups, and then separate time to event models were run for both time periods. And we wanted to ask if environmental variables, including the mean discharge, the minimum discharge levels in a 5-day period, the mean temperature, or the change in discharge and temperature between subsequent 5-day intervals were factors that could cue downriver migrations to start. And we picked these environmental variables because of other studies on green sturgeon further north and some other sturgeon species in general, like lake sturgeon, that have found evidence, variable evidence across populations that these were factors cueing migration. And we developed what's called a hazard ratio, or HR, and this basically indicates the likelihood of an event happening relative to a covariate of interest. So an HR value of greater than 1 meant that there was an increased likelihood of the event happening as that covariate increased. So an HR value of greater than 1 for temperature would mean that with every degree that the water temperature was increasing, it became more likely, that much more likely, that the fish would begin their downriver migration. And an HR value of less than 1 decreased the likelihood of the event happening as a covariate increased. And we used model averaging across multiple competing models to estimate the average effect, the average HR for each of the covariates. And so these were done in 2 separate models. The first panel here is for the early out group. And what really stood out to us was that the minimum discharge was positively related to the likelihood of them beginning the downriver migration. And it actually was the only environmental variable that was significantly related to their movement. We found a similar pattern with the late out migration period, where again, minimum discharge had a smaller but still positive relationship with the likelihood of them beginning to move downriver, but the other variables did not, that were considered. So the take-home message from this was that minimum river discharge rate was positively related to the probability of migration in both groups. Temperature was not related to the probability of downriver migration beginning. And we also wanted to look and try to understand what this meant in the context of what was happening with the water system during this time period. So in California, they measure water years from October to September. So this x-axis actually starts in October of each year, and it goes to the following September. So the early swim down group is actually here on the right, leaving in June. This shaded bar represents that average period for the early departure group. And they were actually leaving as seasonal minimums in flow were approaching. So they would swim upriver here in the middle part of the plot. So you can see the peak flows of the year. That's when they were going against that flow to reach the spawning ground. And then the early swim down group basically seemed to be trying to, the way we were interpreting the result from minimum discharge, they were leaving at a higher minimum discharge during this window. So they tended to be leaving before it reached those lows for the year. And maybe that meant they were leaving before it got so low that it would be increased danger for them swimming through the system. These are long-lived fish that have probably made many migrations over their lifetime. So they may have a certain degree of learning of the patterns of their river system. In comparison, the late down group was leaving right as the first increases in minimum discharge were happening after the summer slow period, essentially. And we were interpreting this potentially as being an indicator that they get those first pulses of flow jumping up in the year, and they go before the flow rates get too high, or before there's something that happens that maybe the flow rates start to drop off again. Because we do see some years where very shortly after the first pulse of flow, it drops off. Another question we had about these green sturgeon, because we were monitoring that each individual could be watched for up to 10 years. We had tags that were transmitting for 10 years. So we wanted to ask if there was repeatability in the outmigration timing across spawning events. So we had multiple migrations for 64 fish. And the day of moving downriver in the first year is plotted on the x-axis, and the y-axis is the first day of outriver migration starting in the second year that they were observed in the river. So these individuals in the two left boxes, or any of the individuals in the two left boxes, they were early in the first year, but 56% of them switched to going down late in the second year that they were observed. And then the two right sides were fish that were late to leave in their first year, and only 20% of those fish moved to leaving early in their second year. We found that there was a non-random association between the early and late groups, so they weren't bouncing around randomly between them. And we think that it's possible that more of the early migrants delayed to the late period based on conditions during the summer months. As I briefly mentioned earlier, California has very recently gone through several years of drought, and it's possible that those drought conditions were cues for fish to hold over until the late period and not move down during the summer months. Overall, conceptual models and the telemetry observations here agreed on the spring upriver migrations, but the downriver migrations occurred over two distinct pulses rather than over an extended time period. And this original conceptual model that we looked at for sturgeon with this gray bar, it's also important to note that the average value of them moving downriver somewhere around September to October, that was actually a period with the telemetry data we saw that they weren't moving. So if we were trying to manage water facilities or fish passage mechanisms for sturgeon in October, we would actually be missing when the population is moving on both sides of things. We think that there are also many questions that remain about green sturgeon in the Sacramento River, including what are the consequences of them spending such long periods of time in the river system? What resources do they need? If a fish is spending almost a year in a river, presumably they're probably doing some sort of foraging. Also, are they at increased risk during this freshwater residency? Are they more vulnerable to disease, to being caught by accident, or just are there possible increased consequences to them for doing this? To sum up this part about sturgeon face a number of conservation challenges, and many are related to gaps in our knowledge about the ecology of these species and even individual populations. There's quite a bit of population variability in this family of fish. Fish passage and connections between marine and freshwater ecosystems are more importantly than we historically perceived, and sturgeon in general are a great example of this. The green sturgeon were migrating 400 river kilometers to reach their spawning grounds. Water flow and other environmental cues are important to helping wild populations persist in natural ecosystems. We often focus on temperature, and temperature is absolutely an important variable for many aspects of life. We can't lose sight of other environmental cues as well, especially when these species may have evolved to pick up very specific environmental cues. If those are lost, it can totally disrupt the life history. Conservation challenges exist across the sturgeon family, and the reason I've got a lake sturgeon over here on the left side is because that is true for lake sturgeon in the Great Lakes region as well. So we are fortunate enough to have lake sturgeon, but they are also facing major conservation challenges in this area of the world. Fortunately, though, we are learning. Acoustic telemetry has proven itself to be a great tool for these animals. And despite the conservation challenges across the sturgeon family, there is still time to help these animals. and to have a positive impact on them. Now I'm going to switch from talking about some of the largest fish in the Central Valley of California to some of the smallest. And this next section is going to focus on Chinook and steelhead smolts that are stocked into the San Joaquin River system. The previous section, actually, that I just talked about on green sturgeon, that is a published paper from Ecosphere. And so if you're interested in the details of that study, you can find it online. It's open access. This part of from here on out, then, of my talk today, this is actually the first group seeing this data. It is work that we are hoping to submit for publication fairly soon, but it is basically you're getting a preview, and there is still some tweaking and refining of data analysis ongoing, but hopefully you can still get some good initial messages from this. So where the Sacramento River system ran north from the Golden Gate Bridge, the San Joaquin River system runs south from the Golden Gate Bridge, but it shares the delta region that we were talking about for green sturgeon. It connects to the Pacific Oceans through that same series of bays and the delta through San Francisco. It does face similar challenges to the Sacramento River, but it also has its own unique systems. Unlike the Sacramento River, there are far more river systems, as you can see in this map, smaller and moderate-sized rivers that flow in and off of the San Joaquin system. So it has its own unique hydrology, and that impacts how fish use it. In the San Joaquin River is home to it's historically been home to a large salmon fishery, including Chinook, which were historically the largest running species in the San Joaquin River. But unfortunately, across California in general, more than 75% of the salmonids are at risk of extirpation or complete extinction for some species within the next century. Steelhead so Chinook were historically the largest run. Steelhead are also present in the San Joaquin River, and they were actually listed as threatened under the U.S. Endangered Species Act. For both species, there are extensive hatchery programs, but much is unknown about the effectiveness of these programs, because once you stock these smolts, it's hard to track them through a system. This is a picture of part of the delta area of the San Joaquin River on the bottom, and you can see there's many channels, there's some lake-like water bodies, so it can be hard to assess what's happening to smolts once they come through here. It also is an area, because of this, that's challenging for the smolts themselves. They must navigate not only the natural variability in the system, but they must also get through and around water reclamation structures, dams, actual pumping facilities drawing on the water directly. They also have to navigate novel habitat patch types that weren't historically present. This lake-like area in the middle of this bottom picture is called Frank's Tract, and it was historically not present. It's there because it's a flooded area to support agriculture in the surrounding areas of this region. This changes the flow dynamics. They evolved to migrate through a river system all the way to the ocean, and now they have to also navigate lake-like water bodies along the way. And there are also numerous predators, native and invasive. There's research going on right now about predation in areas like Frank's Tract, because this is an area where some of the invasives, like largemouth bass, love to hang out and do very well at finding small fish passing through. We've got the research team I was part of came onto this project because we were asked to examine something called species surrogacy. And this is because some management decisions for salmon smolts have been made assuming species can be surrogates for each other. So basically, if we had two salmon smolts of interest, in this case, Chinook and steelhead, we may be doing monitoring efforts that directly assess the success or movements of one of these species and use that to make management plans. Species surrogacy would extend this by saying, well, if we know about this once, if we know about the lighter green species up at the top, we can make assumptions that the orange smolt at the bottom is having the same effects on it. And therefore, we will make management plans for both species based on observations of one. So the major assumption here is that smolts of multiple species share ecological traits such that one species can accurately inform the management and population trends for others. There are some pros to this approach because harder to find species can be inferred from others. So in this case, Chinook, even though they are of conservation concern, they are more readily available than steelhead. And steelhead have the additional hurdles for use and sampling because they are listed as a threatened species. So if species surrogacy can be applied to them, investment in monitoring can be focused on an indicator species like Chinook, and they could basically be used as like a canary in the coal mine of if Chinook are doing well, then we can assume that steelhead are doing well, or if Chinook need this type of management action, so do steelhead. The cons of this approach is that we could be choosing poor surrogates, and then these assumptions break down, and we could actually be it could be quite detrimental to the species that we really want to do well without monitoring directly. So we were asked to assess if Chinook smolts could be used as surrogates for steelhead in the San Joaquin River. So to do this, we wanted to compare the migrations of fish released in the San Joaquin River all the way to the beginning of the bays into San Francisco area. And so if Chinook and steelhead represented full surrogates for each other, they should have similar survival and swimming speeds through the San Joaquin River. And if they represented offset surrogates, so not necessarily identical, but still providing information about each other, then there should be differences in absolute survival and swimming speeds, but they should be predictable and hold across time. So in the San Joaquin River system, there were, much like the Sacramento River, there were receivers throughout the region. We compiled it down to areas that were present every year of the study, so from 2012 to 2016. And then we focused on the main part of the San Joaquin River, so fish that were moving through this main river system only. For this project, fish were tagged with Vemco V4 or V5 180 kilohertz tags that were going off about every 30 seconds. And fish were tagged over a 5-year period. So we filtered the data that was available in the detection files down to those years. We looked only at fish that were released at overlapping release periods. So it had to be the same time of year that fish were released. We only looked at the main San Joaquin River because there were seasonal barriers that were sometimes operated that could influence which way fish could go, but they could always go down the San Joaquin River. And we looked only at fish that were released at a single site that is commonly used for hatchery stocking in this area. And across all years, we looked at over 4,000 each of Chinook and steelhead that had these transmitters. So in terms of survival through the San Joaquin River system, it was relatively low for both species, but it was lower for Chinook compared to steelhead. One thing that stood out to us right away is that almost 40% of fish across both species were not detected following release. So they were put into the river at Durham Ferry, and by the time it was within 5 kilometers downriver, 40% of them were not detected again. And then it continued like that downriver. We did find some indication that survival probability seemed to be related to size, but part of this could also be confounded by the fact that steelhead tended to be larger when they were released. So the steelhead are in blue here on the bottom. You can see that their distribution in sizes tended to be larger than the Chinook. We've also been looking into transit times and forward swimming speed. This is the area that we're still working on, so I'm only going to touch on it a little bit, and this will be fleshed out more, hopefully in the next few months. 603 fish made it to the final array. Chinook were faster. They took 5 days to make it through the whole river down to the bay, whereas steelhead took on average 9 days. We have been working with a linear mixed effects model to try and look at species, year, length, and their interactions and how that relates to the swimming speed as they move through the system. This is the part that's still being developed more, but so far the pattern that we're seeing is that Chinook swam faster for their size than steelhead. There was variability across the years, but not related to the length of those individuals when they were released for either species. So larger Chinook had similar swimming speeds, similar body lengths per second speeds as smaller Chinook. And the same for steelhead. One of the things that we've been dealing with is that within particular years, there can be some sample size issues. You'll see here in this plot, we've ended up dropping to 2013 entirely because the Chinook sample size dropped. They basically disappeared almost immediately after release, and we don't have enough swimming speed estimates to feel we can make reliable measures. One other section to talk about with these fish, going back to the size differences, we did have an area of overlap between them. So rather than just looking at steelhead that were much larger than the Chinook being released, we also wanted to look at what about more closely size matched fish. So the largest of the Chinook and the smaller of the steelhead that were released. And when we looked at the size matched fish from 115 to 200 millimeters total length, we ended up with 670 Chinook and 345 steelhead. And here in this panel, in this figure, each panel represents a detection array from release. So up here on the top left is release, moving down through our array system to the bottom, which is about 3/4 of the way to the bay. We stopped at this point because survival levels got so low that most years we had to drop the consideration. And you can see here that we didn't even consider 2016 because we didn't have any overlapping fish size that year. Survival probability was low for the species, but similar for these smaller size matched fish. About 1% made it to this final array, about 3/4 of the way to the bay. Transit times were still faster for Chinook of the size matched. Chinook took about 6 days, but the smaller steelhead were taking almost 2 weeks if they made it all the way through to the final array. The correlation between the species and in this size match consideration had a trend of being significant across years, and in general there was a 5% offset between the species. Chinook had typically about a 5% lower survival than the steelhead. Just to let you know, we did look at this with all of the fish, regardless of size, and we saw a similar pattern of there being correlations between the two species, but in that case, there was about a 15% offset. The steelhead were roughly 15% more likely to survive to our final array site. Poor smolt survival was not unexpected. This is a vulnerable history of a vulnerable period of life for fish, but it was at particularly high levels, especially for Chinook smolts. There were many years where less than 1% of them made it to the bay, and they still had to get through the bay into the ocean and face predation at all these other steps along the way. This raises questions about could changes in rearing practices improve survival? Also, would it change the relationships between the species that we're describing here? Surrogacy may be extremely limited if use depends on sharing of all traits. So if the species have to be absolutely identical to each other. But relative similarities here indicate that the potential use of surrogates for survival rates is possible in the San Joaquin River. but not transit time. It was clear that Chinook smolts, they get dropped in from the hatchery release and they are booking it through the system quickly. Steelhead are taking a longer period of time to get through, and we're not sure why. Part of it could be because steelhead are not obligated to go to the ocean. They can stay in freshwater and live out their life history as rainbow trout. But most do seem to head that way eventually. We do think that it's worth considering the use of surrogate species in the system, but with care and reevaluation. We shouldn't assume that this will always be a good relationship. If there are changes to hatchery practices, we need to reassess and be very careful before we make these assumptions. But there is the chance that we can use surrogacy to help share the logistical burden of monitoring these fish in this particular system. So you've been really patient, and I'm almost done, I promise. I just want to give a few conclusions before I call it for this talk. Freshwater fish are undoubtedly facing a biodiversity crisis, and this goes from the largest animals to the smallest, and there are new ones that seem to emerge all the time. This top picture is unfortunately from a fish kill of white sturgeon in the San Francisco estuary that happened this year because of a red tide event linked to climate change and nutrient overloading of the system. So we're facing crises that seem to come from all directions, popping up at many different times. Also, it's important to remember that many species perceived as marine, both green sturgeon and the Chinook and steelhead, everything I talked about today would often be perceived as marine. They tend to spend most of their lifetime in the marine system, but they are absolutely dependent on access to freshwater ecosystems. We can't overlook the access to freshwater, or we can't support conservation of these species. Fortunately, the breadth and depth of our ecological knowledge is rapidly growing. One of the tools for that has been acoustic telemetry. It is a tool that is being pursued in increasing application to aid management assessments and decisions. Although it is important to remember it's not a panacea. It's not going to be the right answer or the right tool all the time, but it is a tool in our kit. And finally, we face many challenges ahead, but fortunately, we have some of the best tools at our disposal yet to make a difference. And I'm just trying to leave on a message of don't be, I'm not a defeatist about the freshwater biodiversity crisis and challenges we have, because I think we have the best tools and opportunities right now to try and make a difference. And with that, I'll say thank you for your attention, and hopefully we have a few minutes for some questions if you have some. I've got my contact information up here too, so feel free to reach out later.
Great. Thank you so much. That was an awesome talk. And also thanks for sharing with us too some data that a lot of people haven't seen yet or is still kind of in review. That's been really awesome to see. So I encourage people right now, if you have any questions, feel free to put them in the chat, or you can like raise your hand, unmute yourself. But yeah, we do have a few minutes for questions. We'll give people a second. Sometimes you have to take a bit to type up your message in the chat. Yeah, go ahead.
Yeah, I'm wondering if you have any concerns about marker influences on survival for the acoustic tags.
Oh, yes. With the sturgeon, we don't. The tags are such a small burden compared to their body size. Also, the vast majority of the fish, more than 95% of them, are detected over almost the entire 10-year period after they've been released. So we don't, with sturgeon in particular, we don't. There are some concerns with the smolts, because it's pushing the typical recommended body size limits for the acoustic tags. These fish were done with a subset held in a hatchery for a period of time with dummy tags. And the surgery technique used on these particular smolts, I believe it was a less than 10% mortality rate in the month following tagging. And also most of the mortality happened within the first 48 hours. And these smolts were held for almost a week after tagging before they were released. But that was a very good point to bring up because it is something that should be considered in designing, especially with the smaller bodied fish.
I think that also kind of aligns with the question that was in the chat from Bridget, that it's something to do with significant mortalities due to telemetry implants. So I think that also kind of answered her question as well. Bridget, if it didn't, you can always send another message or say something as well.
Thank you for bringing that up, though, for both of you bringing that up, because I think it's often overlooked. There's kind of this perception of we'll tag a fish and it's ready to just go.
I'll ask a follow-up if that's okay. And I know in the Columbia River, there's a lot of concern with avian predation on migrating smolts, and they're using acoustic tags too. And I'm just wondering if there could be subtle differences in vulnerability to avian. First of all, maybe does avian predation seem to be an important source of mortality for smolts in the San Joaquin? And is there any possibility that tags might influence behavior in a way that would exacerbate that?
That's also a really interesting question. And the lab that I was part of with UC Davis, they're actually there's some grad students now trying to get further into that. They know that there's significant fish predation. They're actually doing some experiments right now where they have smolts that are tethered to a reader that detects when they're plucked off by another fish, basically. And but there is definitely the possibility of avian predation. And in terms of if the fish with tags are more likely to be vulnerable, that's something that needs further work, because we don't know if it's slowing them down enough that it could maybe increase the risk, or if it may influence them to swim higher up. For example, so that is an area that I think needs future study and further consideration.
All right. Anyone have any other last minute questions? Again, feel free to unmute. Last anything in the chat. Otherwise, if we don't have anything that comes in, I just want to say again, thank you so much for giving a talk. That was really interesting, and we really appreciate too, especially seeing a freshwater side of talks here. So with that, we'll say thank you, and that'll conclude our seminar for this week.